learning experience. You have recorded classes where you can access your subjects anytime, anywhere as per your schedule. Then you have notes based on the latest CDME syllabus. And apart from that, we have MCQs to cater to everybody's need, beginning from beginners level to the expert level, where we have standard multiple choice questions. And apart from that, we have personalized counseling sessions where you can book a slot with us anytime for any extra guidance or support that you need, apart from the academic support. Then we have 3 different subscription plans at Auricle, starting from basic, which is for 3 months. You can avail it for 15K. Then you have the standard plan, which is for 6 months, available at 30,000. And then you have the mentorship plan, where you have live doubt clarification sessions and one-to-one personalized guidance, which will be available for 50,000 and will last you for 12 months. You can also get additional 20% off on any of the subscription plans as mentioned above by using my promo code displayed on the screen here. Then in this internal exam marathon series, we are aiming to give you a rapid revision of all the important topics which are most frequently asked. We also be discussing about volatile facts. Then we have multiple choice questions to assure you top-notch grades in your exams. So this series is specifically oriented for your university exams for all these states. So before taking any more time, let's begin with fatty acid synthesis. So beginning with the introduction, as we always do, that when does fatty acid synthesis takes place in the body? So it takes place in the well-fed state. So the hormone of the well-fed state, as we all know, is insulin. Okay, so your fatty acid synthesis is taking place in the well-fed state under the hormone insulin. Then fatty acid synthesis, that whatever fatty acid has been synthesized in your body, it is stored as TAG in adipose tissue. So what is TAG? The fatty acid that has been synthesized, it combines with glycerol to form tags. So your fatty acids that have been synthesized, they are stored as tags in adipose tissue majorly. Then palmitic acid is the most common fatty acid that is synthesized in your body. Okay, yeah. Then site, it is synthesized in liver, adipose tissue, kidney, brain, and mammary glands. Then the organelle that is involved is the cycle is extra mitochondria. That is, it is a cytoplasmic cycle. It is also known as Leyland spiral based on the person who first elucidated it. Then this pathway, it needs energy, okay? So just like gluconeogenesis, where you were synthesizing glucose, ATP was needed. Similarly, when you're synthesizing fatty acids, ATP is needed, okay? This is not an energy producing pathway. This is an anabolic pathway. The starting material is acetyl-CoA, and the source of acetyl-CoA is the link reaction and the beta oxidation. So what is the link reaction? If you remember, in aerobic glycolysis, what was formed in glycolysis? Pyruvate was formed, okay? And in aerobic glycolysis, when there is oxygen, that pyruvate via the link reaction will form acetyl-CoA. The enzyme is pyruvate dehydrogenase, okay? And in beta oxidation also, what were you getting? Acetyl-CoA. So the source of acetyl-CoA is the link reaction and the beta oxidation, which takes place in the mitochondria. So acetyl-CoA has to be transported to cytoplasm for fatty acid synthesis. Like I just said here, it is an extra mitochondrial pathway. So, but your acetyl-CoA, which is the starting material, it is present in the mitochondria. So our first step will be to transport that acetyl-CoA from the mitochondria to the cytoplasm. If you remember, day before yesterday when we were studying beta oxidation, what were we doing? we were transporting the activated fatty acid from the cytosol inside the mitochondria. Okay. Now moving ahead, the three basic steps of fatty acid synthesis. So the first one is transfer of acetyl-CoA from the mitochondria to the cytoplasm. The second is acetyl-CoA carboxylase reaction. So this is a carboxylation reaction, and this is a biotin dependent carboxylation. And the third step is fatty acid synthase complex reactions, that is FAS reactions. Okay, now we'll take each of these steps one by one in detail. So the first step being the transfer of acetyl-CoA that is present in the mitochondria from the mitochondrial matrix to the cytosol. So this is our first step. Now your oxaloacetate is a 4-carbon compound and your acetyl-CoA is a 2-carbon compound. So a 4-carbon compound, that is oxaloacetate, is going to combine with this 2-carbon acetyl-CoA, and what is formed? Citrate is formed, and your citrate will be 6-carbon 4 and 2 equals to 6. So a 6-carbon citrate is formed, and this citrate is a tricarboxylic acid. So this citrate, which is a tricarboxylic acid, it has a transporter for itself in the inner mitochondrial membrane known as the tricarboxylic acid transporter. Okay, so this tricarboxylic acid, it has a transporter in inner mitochondrial membrane, which is known as tricarboxylic acid transporter. So this citrate via this transporter will be transported from the matrix into the cytoplasm. Okay, now your citrate has reached the cytoplasm. Now in the cytoplasm, it will be acted upon by another enzyme, that is citrate lyase. And this is an ATP dependent enzyme. So what will happen? An ATP is required, a water molecule is also added, and your citrate is split into, again, oxaloacetate and acetyl-CoA. So this way you can see your acetyl-CoA has been transported from the matrix into the cytosol. Okay. So that was the first step. Now moving on to the second step, that is the reaction of acetyl-CoA carboxylase. Now this acetyl-CoA that has been transported into the cytoplasm, to this, what is added? A carbon dioxide molecule is added. Okay, and who is the donor of this carbon dioxide? This carbon dioxide is donated by a carbonate ion. Okay, so this 2-carbon acetyl-CoA is changed into a 3-carbon malonyl-CoA. Okay, so a carbon is being added. So when a carbon is being added, what it is? It is a carboxylation reaction. So in carboxylation reaction, what is happening? A 2-carbon compound is changed into a 3-carbon compound. Okay, and this is a biotin dependent carboxylation, and ATP is also required. You can see one ATP is changing into ADP. So that was the second step of fatty acid synthesis. Now most of the fatty acid synthesized in our body, the most common fatty acid, okay? Most common fatty acid synthesized in our body is palmitic acid. So very long chain and long chain, other very long chain and long chain fatty acids, they are elongated on this palmitic acid. Very long chain fatty acid are elongated on this. Okay, if you remember during beta oxidation, I told you that the most common fatty acid that undergoes beta oxidation is your palmitic acid, and it is also the most common fatty acid that is synthesized in our body. That is also palmitic acid, and this palmitic acid is only 16 carbon long. So the other long chain and very long chain fatty acids, they are elongated on this palmitic acid. Okay, now moving on to the reactions of FAS. This is the longest phase in fatty acid synthesis. Longest phase in fatty acid synthesis. Now, what is happening in FAS? What is FAS actually? This FAS complex, it is a homodimer. A dimer means it has two polypeptide chains, okay, and homo means same. So it has two similar polypeptide chains, okay. So this FAS is a homodimer, and each monomer of this FAS has six enzyme activities, and one acyl carrier protein. As you can see here in this diagram, the primary structure, you can see 1, 2, 3, 4, 5, 6. 6 enzymes and one ACP, acyl carrier protein. Okay, now this ACP has vitamin B5 in the form of 4-phosphopantothene. That is, it has pantothenic acid, and in X-ray crystallography, it has been seen that it is in the shape of an X. You can see this. It has an X-shaped structure. This you can see. The FAS is a multifunctional enzyme. That is, a single polypeptide chain has more than 2 enzyme activity. And how many enzyme activities are there? 6. This FAS has 6 enzyme activity. So, like a bifunctional enzyme, it is an enzyme which has it is a single polypeptide chain which has 2 enzyme activity. But this FAS is not a bifunctional enzyme, it is a multifunctional enzyme. That is, this single polypeptide chain, it has more than 2 enzyme activities. That is, it has 6 enzyme activities and one acyl carrier protein. So in each monomers, what is happening is that there are 3 domains, okay? So the first domain, it is the condensing unit, the second domain is the reduction unit, and the third domain is the ACP unit and the acyl carrier protein. You can see here these are arranged into domains. So the first domain has 2 enzyme activities, that is malonyl or acetyl transacylase and ketoacyl synthetase. So this is the first domain and it has 2 enzyme activities. Earlier and older editions of Harper, if you see, these are present, that is acetyl and malonyl transacylase. It is given as 2 different enzyme activities, but now they're included in one. Now the second unit, that is the reduction unit, forms the second domain. It has 3 enzyme activities, that is ketoacyl reductase, dehydratase and enoyl reductase. So they form the second domain. This was the first domain, and the last domain, that is the releasing domain, it has ACP and the thioesterase. This is the last third domain. Okay. Now, before we move forward to the reactions of FAS, let us take this MCQ. So it says, which of the following is not a part of fatty acid synthase complex? That is, it is not a part of FAS. So ketoacyl reductase, yes, this is a part of FAS. It is present in the second domain. Enoyl reductase, it is also present in the second domain. So it is also a part of FAS. Then acetyl-CoA carboxylase. No, this is not a part of FAS. Then ketoacyl synthase is also a part of FAS. This is present in the first domain. So our answer will be acetyl-CoA carboxylase. This is not a part of FAS. Okay. So before we move on to the reactions of FAS, let me quickly recapitulate what all we've done. So what are the important points that you must write if you're asked a question on fatty acid synthesis? That is the site, it is extra mitochondrial, that is taking place in the cytoplasm. Okay. Then what else do you need to write? The steps. What are the steps? The first is that is the transport. Transport of what? The starting material, the acetyl-CoA from the mitochondria to the cytoplasm. The second being the reaction, that is the carboxylation reaction, acetyl-CoA carboxylase reaction. And the third is the reactions of fatty acid synthase complex. So now let us talk about the reactions of FAS. So as we've said that FAS has two domains. So for the sake of convenience, so that you can easily understand, you can draw this. This diagram might look very complicated. So I'll give you an easier version. Okay, first domain, second domain. So now it says it is your FAS is a homodimer. That is, it has two similar polypeptide chains. So I've drawn these two similar polypeptide chains here. Okay. Now in the first monomer, the cysteine residue of the first monomer, we all know the cysteine has a sulfhydryl group. Okay. And in the second monomer, actually, let me draw it like this. Like this, because they are present on two different monomers. Okay, we're talking about two different monomers. So let's draw it like this. So the sulfhydryl of cysteine in the first monomer and the sulfhydryl of ACP in the second monomer. Let's denote it like this. Okay, now what is happening here? See, your acetyl-CoA, it first comes and attaches to the sulfhydryl of ACP, and then it shifts to the sulfhydryl of cysteine. So basically what is happening, your acetyl-CoA is attaching to the sulfhydryl of cysteine, but this is taking place in two steps. But for the sake of convenience in the examination, you need not go in so much detail. You can directly say what is happening, that acetyl-CoA comes and attaches to the sulfhydryl of cysteine. Okay. So when it will come and attach, what will happen? Okay. Or simply you can write it in words. That is, to this your acetyl group comes and attaches, which is 2 carbon. Okay. And to the ACP, what comes and attaches? Your malonyl comes and attaches, which is 3 carbon. Okay. So in the first step, and this is mediated by which enzyme? This is mediated by the first enzyme, that is your acetyl malonyl transacylase. Okay, let me repeat this for you. I don't know why my highlighter is not working. Just give me a second. Okay. Okay. So let me repeat this for you. What is happening in FAS? Your FAS has two monomers. So I'm saying to the first monomer, in the first monomer, there is a cysteine residue. So we all know that cysteine is a sulfhydryl group. So to the sulfhydryl of the cysteine in the first monomer, acetyl groups comes and attaches. Okay. Here. And to the sulfhydryl of ACP in the second monomer, your 3 carbon malonyl groups comes and attaches. Okay. So we've reached this stage. Here you can see to the sulfhydryl of ACP, what has been attached? Your malonyl. And to the sulfhydryl of cysteine, what is attached? Acetyl is attached. Now what happens? There is a decarboxylation reaction taking place from the malonyl. Okay. So to the malonyl, that carbon dioxide which was added by the acetyl-CoA carboxylase, that is removed. Okay. So decarboxylation is taking place in the second step. and a carbon dioxide is removed. Which carbon is removed? The one which was added by acetyl-CoA carboxylase, the carboxylation reaction, the second step that had taken place. Okay. Now what happens? Your acetyl group condenses with the malonyl at ACP. You can see this. This carbon dioxide has been removed and this comes in, attaches here. You can see your acetyl condenses with malonyl at ACP. Okay. Now what is formed? A 3-keto acyl group has been formed. 3-keto acyl group has been formed. You can see here. There's a keto here at the third position. So a 3-keto acyl group has been formed. Now the next 3 steps will convert this 3-keto acyl group to its corresponding saturated acyl group. Okay. So in the next 3 steps, 3-keto acyl group is changed to its corresponding saturated acyl group. So how do you bring that about? That is this keto group is first changed into hydroxyl, you can see here. And this hydroxyl is then changed into a double bond between alpha and the beta. And then you can see this double bond is changed into a single bond. And then you end up with butyryl ACP. That is a 4-carbon saturated fatty acid. Okay. And how is this taking place? Let's see now. This keto group here, via the action of the enzyme keto acyl reductase. Okay. So this enzyme is going to act here, keto acyl reductase, and what will happen? The keto group is reduced to an alcohol. You can see there was a keto group here and there's an alcohol group here. So your keto group reduced to hydroxyl group. Now what is happening? Water is removed. You can see here, water is being removed to create a double bond. Water is removed to create double bond between alpha and beta carbon. You can see a double bond here between alpha and beta carbon. See this? This is double bond here between the alpha and the beta carbon. This. I hope it's visible. Then this double bond is reduced to a single bond. You can see here. This single bond here. So your double bond reduced to a single bond. So these are the 3 steps we were talking about, that how your keto acyl group has been converted to its corresponding saturated fatty acyl group. So you end up with 2 acetyls were condensed. So you end up with what? A 4-carbon butyryl. Now what happens? This 4-carbon butyryl again shifts to the sulfhydryl of cysteine. Now a new malonyl CoA comes and it attaches to the sulfhydryl of ACP. Then again from that malonyl CoA, what will happen? Decarboxylation will take place. Now what will happen? There will be condensation in ACP and the butyryl condenses with the malonyl after decarboxylation at ACP. Now again, these 3 steps will take place. That is your keto being reduced to alcohol. Then from the alcohol group, a double bond is created and then double bond is reduced to a single bond. And then you end up with a 6-carbon saturated fatty acyl ACP. So this way, this cycle will continue until you end up with those many number of carbons as long as the fatty acid that you are synthesizing. Here in this case, 16 carbon because we are synthesizing palmitate. Okay. So these were the 3 basic steps involved in fatty acid synthesis. So the minimum that you need to write if you get a question on fatty acid synthesis is, firstly, We need to mention about the transport of acetyl-CoA from mitochondria to the cytosol. So for this, you have to mention how citrate is being formed, and then you have transporters for citrate, and then again that citrate is being lysed into oxaloacetate and acetyl-CoA in the cytoplasm, and this way your acetyl-CoA has been transported into the cytoplasm. Then the second step is your formation of malonyl-CoA, that is the carboxylation reaction brought about by acetyl-CoA carboxylase. And then the third is the fast reactions. Okay, and you also need to know a little about fast, because sometimes you might get asked only about fast complex and short notes. Write a short note on fast complex. Okay, so that fat is a homodimer. It is made up of two polypeptide chains which are similar. Then each monomer has 6 enzyme activities, and it is an ACP. It has an ACP. And then you move on to the reactions of fast. Okay, reactions of fast. If this diagram is very complicated, then you can change it to this one, like I've shown you here in this image. You can denote your fast like this with the help of these two curved lines. Okay, so what is happening in the first step? To the sulfhydryl group of cysteine in one monomer, what comes in attaches? Your acetyl group comes in attaches, and to the sulfhydryl of ACP in the second monomer, your malonyl group comes in attaches. Then via decarboxylation, a carbon dioxide is lost from malonyl group. Then what happens? Your acetyl-CoA condenses with the malonyl at the ACP. Then you end up with 3-keto-acyl group. Okay, and the next 3 steps, you are just changing that keto group into its corresponding saturated acyl group. That is, the keto group will be reduced to its to a hydroxyl, and then water is removed to create a double bond, and then that double bond is reduced into a single bond, and then these steps will again be repeated. Now moving on to the regulation of fatty acid synthesis. So there are 2 types of regulation: short-term and long-term regulation. Short-term regulation is very short-acting and it is fast. It consists of allosteric regulation, covalent modification, and compartmentalization. And long-term regulation, if the body has enough fatty acids synthesized, then what will happen? There will be decreased expression of enzymes that synthesize fatty acid. So the enzymes that are synthesizing fatty acid, their expression will be decreased in case the body is already having a lot of fatty acid. So the important short-term regulations, they are more important. Let's talk about them. That is your allosteric regulation. So acyl-CoA carboxylase is the rate-limiting enzyme. Now your acyl-CoA carboxylase, it is favored by citrate and inhibited by long-chain fatty acid. So if there are already, as we've said, if there are already fatty acids present in the body, they have been synthesized, what will happen? Your acyl-CoA carboxylase, which is the rate-limiting enzyme, it will be inhibited by those fatty acids. And citrate is transporting the starting material acetyl-CoA. So what will happen? If there is a lot of citrate, it will favor fatty acid synthesis by favoring what? By favoring the rate limiting enzyme, that is acyl-CoA carboxylase. And also your citrate is an activator of TCA transporter as well. Okay, so if the transporter is activated, more citrate will be transported from the matrix, mitochondrial matrix, into the cytoplasm for fatty acid synthesis. And vice versa, if there are more long chain fatty acids, the transporter, TCA transporter, is inhibited. And also, how is your this rate limiting enzyme, that is acyl-CoA carboxylase, activated in its inactive form? It exists in the form of a dimer, as you can see. And but when it is activated by citrate, what happens? It polymerizes into its active form. Okay, when it forms a polymer, it becomes activated. When it is inactive, it exists in the form of dimer. Now, covalent modification is brought about by insulin, as we've discussed, that your fatty acid synthesis takes place in the well-fed state. So obviously it will be under the influence of the hormone insulin. And insulin, we all know that it dephosphorylates enzyme. So fatty acid synthesis will be under the influence of insulin as it takes place in the well-fed state, and acetyl-CoA carboxylase is active in dephosphorylated state. So you can see here, when phosphorylation is taking place, it becomes inactive, that is, it exists in the form of dimers. And when dephosphorylation takes place, then it polymerizes and exists as active polymers. You can see here, your insulin, it favors what? It favors protein phosphatases. And what do phosphatases do? They remove phosphate groups. Okay. So from acetyl-CoA carboxylase, which was phosphorylated, it will remove this phosphate and change into acetyl-CoA carboxylase. This dephosphorylated acetyl-CoA carboxylase is active. And what happens if glucagon and epinephrine are there? They will activate kinases, and kinases, they will phosphorylate this acetyl-CoA carboxylase, and what will happen? It will become inactive. So that was about covalent modification. Now another type of regulation is compartmentalization. As we all know that beta oxidation was taking place in the mitochondria and fatty acid synthesis in cytoplasm. So this compartmentalization also helps in regulation of both fatty acid synthesis and beta oxidation. So the two pathways are separated from each other with the help of a compartment which is impermeable to each other. As we see that the outer mitochondrial matrix is almost permeable to most of the compounds, but the inner mitochondrial matrix is impermeable. Now moving on to the elongation of fatty acids. So the fatty acid synthesizes only 16 carbon, but body needs longer fatty acids, especially your brain. Therefore, elongation of fatty acids takes place. Okay. Now, where does it occur? It occurs in smooth endoplasmic reticulum and in mitochondrial fatty acid elongase. So, in the endoplasmic reticulum, an enzyme called microsomal fatty acid elongase system. This is what is helping in the elongation of fatty acid, and this is the major site of elongation. And mitochondrial fatty acid elongase system, that is the minor site of elongation. Now, synthesis of unsaturated fatty acids, there are two enzymes required, elongase and desaturase, and the site is endoplasmic reticulum. What does desaturase do? It removes the hydrogen and makes saturated fatty acid unsaturated. So, it removes hydrogen and makes saturated fatty acid unsaturated. So that was about fatty acid synthesis. So the minimum that you need to know in fatty acid synthesis is at least the site, the organelle, that is site, liver, adipose tissue, kidney, brain, mammary gland, the organelle that is take place in the cytoplasm, and the three basic steps of fatty acid synthesis, that is the transport of acetyl-CoA from the matrix, mitochondrial matrix, into the cytosol. Then second is the carboxylation reaction brought about by acetyl-CoA carboxylase, and third is the reaction of Fas. And you also need to know about the Fas complex because sometimes you get that in short notes. So this is like the minimum that you need to know in fatty acid synthesis. Now moving on to our next topic, that is cholesterol synthesis. Okay, so cholesterol, if you break up this term, chole, it is something related to bile, and sterol because there is a sterol ring present in the structure of cholesterol. Okay, so that is the breakdown for the word cholesterol. Now, cholesterol is used for anabolic purposes, and it is not used for energy generation purpose in the body. Okay, so you will use cholesterol for the synthesis of vitamin D, steroid hormones, etc. But there is no energy production role or energy generation role of cholesterol. Now, cholesterol synthesis also takes place under the influence of hormone insulin, just like your fatty acid. Once inside the body, cholesterol can only be excreted as bile acid. So the only excreted form of cholesterol is bile acids. That is how the body can get rid of cholesterol. So that was a brief introduction about cholesterol. Now moving on to the structure of cholesterol. It consists of, although the structure is not very important, but we talk about the structure for the sake of MCQs. Okay. And theory questions as such, the structure is rarely asked. But you need to know, like, it has the name of the ring that is present, that is cyclopentano per hydrophenanthrin ring. What is that? Your cholesterol, it consists of a 4 fused hydrocarbon rings. You can see the structure here. There are 4 fused hydrocarbons ring A, B, C, D. Okay, called the steroid nucleus. So these 4 fused hydrocarbon rings, they together form the steroid nucleus. And the name of this ring is known as cyclopentanoperhydrophenanthrin ring. So sometime in MCQ can be the ring present in the structure of cholesterol, and you have names of different rings. So your answer will be this. Now it has a 8 carbon branch hydrocarbon chain attached to the carbon 17 of D ring. You can see this is the D ring. To its carbon 17, what is attached? A hydrocarbon tail has been attached, which is a 8 carbon branched chain. Now to the ring A, there is a hydroxyl group present at the 3rd carbon. And there is also a double bond you can see here in B ring between carbon 5 and 6. Now cholesterol in total has 27 carbons. Okay. The steroids with 8 to 10 carbon atoms in the side chain at carbon 17 and a hydroxyl group at carbon 3 are classified as sterols. Now, all sterols are steroids, but not all steroids necessarily be sterols, okay? Because only those steroids which have a hydrocarbon chain at carbon 17 of D ring and a hydroxyl at ring A at the 3rd position, they classify to be sterols. Now, your cholesterol is amphipathic. That is, it has both polar and non-polar ends. But what happens when it forms a cholesterol ester? That is, it combines with the fatty acid and is engaged with it in an ester linkage. It becomes what? It becomes hydrophobic. Because the hydroxyl which was providing it polarity is no longer there. It is engaged in an ester linkage. Now, moving on to the sites of cholesterol synthesis, it is synthesized by almost all tissues in humans, maximum contribution being made by liver, intestine, adrenal cortex, reproductive tissues like ovary, testes, and placenta. So this is the site of cholesterol synthesis. Organelle is, it is both synthesized in the cytoplasm and the smooth endoplasmic reticulum. So these are the organelles involved. Now, these steps of cholesterol synthesis, this is what is most important. That is the steps of cholesterol synthesis. This is the minimum that you need to know. So what happens is 2-acetyl-CoA, it combines together, and the enzyme is acetoacetyl-CoA to form what? Acetoacetyl-CoA synthase is the enzyme and forms what? Acetoacetyl-CoA, which is 4-carbon compound. Now, in ketone body synthesis, which we'll be talking about in our coming lectures, so you will see that this is also an enzyme forming in ketone body synthesis. So 2-acetyl-CoA, they combine to form acetoacetyl-CoA, and the enzyme is acetoacetyl-CoA synthase. Now what happens? This acetoacetyl-CoA, it fuses with another acetyl-CoA to form HMG-CoA, which is beta-hydroxy beta-methyl-butyryl-CoA, and the enzyme involved is HMG-CoA synthase. Okay, Now, this is also common in both ketone body synthesis and जो cholesterol synthesis. Now, this HMG-CoA, which has been formed, it has three fates. That is, it can go for cholesterol synthesis, it can also go for ketone body synthesis, and it can also go for leucine catabolism. Now, if there is an MCQ where the question is something like which of the following enzymes is common to both ketone body and cholesterol synthesis, okay? So in if you have both the options available, that is acetoacetyl-CoA synthase and HMG-CoA synthase. So your best option will be HMG-CoA synthase. But in a question where HMG-CoA synthase is not given, then you're going to mark acetoacetyl-CoA synthase. Now what happens? This HMG-CoA synthase, which is acted upon by HMG-CoA reductase, which is the rate limiting enzyme to form mevalonate. Okay, this mevalonate is 6-carbon low. Now, one important clinical application that you need to know here, that statins used in hypercholesterolemia, they are competitive inhibitors of this HMG-CoA reductase. So, statin drugs, they are used where? In hypercholesterolemia. They are competitive inhibitors HMG-CoA reductase. Okay. Now what happens? This mevalonate, it undergoes a series of phosphorylation and decarboxylation reactions. Okay. Now these phosphorylation reactions, they happen in 3 steps. You can see step 4 is of 3 parts, A, B, and C. So to this mevalonate, first a phosphate, you can see an ATP is involved here, is added and forms 5-phosphomevalonate. Then another phosphate is added and forms 5-pyrophosphomevalonate. Then a third phosphate is added and forms 5-pyrophosphomevalonate. But if you cannot memorize this much, then you can also skip this and you can simply write mevalonate. See, acetyl-CoA, two acetyl-CoA, they combine to form acetoacetyl-CoA. Then that acetoacetyl-CoA again combines with another acetyl-CoA to form HMG-CoA. Okay, now this HMG-CoA, it is acted upon by HMG-CoA reductase and forms mevalonate. Now, if you cannot remember all the 3 steps, then you can simply write this mevalonate. It undergoes a series of phosphorylation and decarboxylation reactions. You can see here kinase, and decarboxylase being, and you end up with what? Isopentenyl pyrophosphate. So you end up with an isoprenoid unit, which is 5-carbon long. Okay, because the decarboxylation has taken place, a carbon dioxide is removed. So your 6-carbon mevalonate, it changes into an isoprenoid unit, which is 5-carbon. Now this 5-carbon isoprenoid unit, this is the building block of cholesterol. This here, this is the building block of cholesterol. So this isoprenoid unit, they are going to join up to form the 30 carbon squalene in stepwise manner. Now let's see how. Firstly, this isopentenyl pyrophosphate, it undergoes isomerization to form dimethyl allyl pyrophosphate. Again, this step is not very important. You can simply say two isoprenoid unit, they fuse together to form 10 carbon geranyl pyrophosphate. Okay, so you can just simply show in this step that another isoprenoid unit is coming to form your geranyl pyrophosphate. So these are 5 carbon, so this will be 10 carbon. Okay. Okay. So you can see what is formed. Your 10 carbon geranyl pyrophosphate is formed. Now to this 10 carbon, another isoprenoid unit, which is 5 carbon, is being added. So what you end up with? You end up with a 15 carbon farnesyl pyrophosphate. Now two farnesyl pyrophosphate, they fuse together to form this 30 carbon squalene, because farnesyl pyrophosphate, it is 15 carbon, two of them fusing. So you get this 30 carbon squalene. Now this squalene, via the enzyme epoxidase, it changes into squalene epoxide, then forms lanosterol, zymesterol, desmosterol, and ultimately you get your 27 carbon cholesterol. And this lanosterol is the first cyclic compound that is being formed. First cyclic compound that is formed in cholesterol synthesis. So that was about the steps of cholesterol synthesis. So these steps are important. I'll just recapitulate in brief. So your 2-acetyl-CoA via the action of aceto-acetyl-CoA synthase, they fuse together to form what? Aceto-acetyl-CoA. Now this aceto-acetyl-CoA again fuses with another acetyl-CoA, and the what is formed? HMG-CoA is formed, and the enzyme is HMG-CoA synthase. Now, HMG-CoA is acted upon by the enzyme HMG-CoA reductase, which is also the rate-limiting enzyme. One clinical is that you statin drugs that are used in hypercholesterolemia, they are also competitive inhibitors of HMG-CoA reductase. Okay, so if there is a hypercholesterolemia, already the cholesterol level is high, so you give statin drugs which will inhibit HMG-CoA reductase. That is the rate-limiting enzyme of cholesterol synthesis pathway. So what will happen? Your cholesterol synthesis will not take place. Then this HMG-CoA reductase, it forms what? It forms mevalonate. Then this mevalonate will undergo phosphorylation or decarboxylation to end up with a isoprenoid unit, which is 5 carbon. Then two isoprenoid units, they fuse together to form a 10 carbon geranyl pyrophosphate. Then to this geranyl pyrophosphate, another 5 carbon isoprenoid fuses to form farnesyl pyrophosphate, which is 15 carbon. Then two farnesyls, they condense together to form 30 carbon squalene, and then this squalene ultimately, after forming the intermediates lanosterol, zymosterol, desmosterol and all, to end up with cholesterol. So this is the simplest way how you can write about the steps of cholesterol synthesis. If you learn the products in the substrate with the help of carbons, it will be very easy for you to remember. Okay. Now let's take an MCQ. Enzyme common for synthesis of both ketone bodies and cholesterol. So HMG-CoA reductase, HMG-CoA synthase, acetyl-CoA carboxylase, and HMG-CoA lyase. So your answer will be HMG-CoA synthase. This is the enzyme common you can see here. HMG-CoA synthase. Okay. Now moving on to the functions of cholesterol synthesis. 50% of the cholesterol that is formed, it is excreted as bile acids through bile. And the rest is used for vitamin D synthesis and steroid hormone synthesis. Your steroid hormones like sex hormones and corticosteroids. And your vitamin D, which is derived from 7-dehydrocholesterol, okay, that is converted into vitamin D. Taking the MCQ here, cholesterol is a precursor of all except bile salts, bilirubin, steroids, and vitamin D. You can see here your bilirubin is the answer, as steroid hormones, vitamin D, they are synthesized from cholesterol. Your bile salts, they are also synthesized from cholesterol. We will see how cholesterol from bile acids and then bile salts are also formed from it. Okay, so your answer will be bilirubin here. Now moving on to the regulation of cholesterol synthesis. There are three types of regulation: feedback regulation, feedback inhibition, and hormonal regulation. So feedback regulation, if there is enough of dietary cholesterol, then this decreases binding of SREBP at genes that synthesizes HMG-CoA reductase. Okay, so it is saying when already there is enough cholesterol in the body through diet, then what will happen? The HMG-CoA reductase will not be synthesized. Okay, why? Because there will be decreased binding of the protein at the genes that synthesize HMG-CoA reductase. So this leads to decreased expression of genes that synthesize HMG-CoA reductase, and we all know that it is a rate limiting enzyme, and it is a long-term regulation which acts at genetic level. Okay, so your feedback regulation is taking place at the genetic level, where your steroid regulatory element binding proteins, they will not bind to that gene which synthesizes your rate limiting enzyme HMG-CoA reductase. Feedback inhibition, this acts as protein level only and not at gene level. Increased mevalonate, it inhibits HMG-CoA reductase. So what is happening here in this case? So HMG-CoA, when it was acted upon via the enzyme HMG-CoA reductase, it forms what? It forms mevalonate. Now, when this mevalonate is increased, it will inhibit this enzyme HMG-CoA reductase. Okay, so this is feedback inhibition where the product is inhibiting the enzyme. So this is taking place only at the protein level and not at the gene level, whereas feedback regulation takes place at gene level, where the expression of the gene which is synthesizing the enzyme is only decreased. A hormonal regulation, as we know that cholesterol synthesis takes place in the well-fed state, so it will be under the effect of insulin. So insulin dephosphorylates the rate-limiting enzyme HMG-CoA reductase and thus activates it. Okay, the cholesterol synthesis is an anabolic pathway. And we know anabolic pathways are under the influence of insulin. So insulin increases cholesterol synthesis. So moving on to bile acids. Bile acids are the excretory form of cholesterol. So the only way to excrete cholesterol from our body is to convert it into bile acids. Now the bile acid pool is highly regulated in the body. The total bile acid in the body is increased. Then bile acid synthesis is decreased. A bile acid is synthesized in two sites, that is your liver and the intestine. In the liver, what is happening? Bile acids formed in the liver, they are known as primary bile acids. Cholesterol is first hydroxylated via the enzyme 7-alpha hydroxylase to form hydroxycholesterol, which through multiple steps form two primary bile acids, that is cholic acid and chenodeoxycholic acid. So you can see in the liver what is happening. This is your cholesterol. It is acted upon by an hydroxylase enzyme to form what? It forms hydroxycholesterol. And for this reaction also what is needed? NADPH, oxygen and vitamin C is needed. If you remember, when we were studying about HMP shunt, we had also studied that NADPH that is produced in the HMP shunt, it is needed for reductive biosynthesis of fatty acids and cholesterol. So this is the step where it is required. NADPH is required. Now this hydroxycholesterol that is formed through multiple steps, it ultimately ends up forming what? Primary bile acids, that is cholic acid and chenodeoxycholic acid. Now, these primary bile acids that are formed in the liver, they are conjugated by glycine and taurine. Okay, so these primary bile acids that are formed, they are conjugated by glycine and taurine to form what? Glycoproline and tauroproline. Now the conjugated bile acids, they are excreted through the common bile duct into the intestine. So now the further metabolism of these primary bile acids takes place in the intestine. So they are then excreted, these conjugated via common bile duct into intestine for further metabolism. Okay. Now let's see what is happening in the intestine to these primary bile acids. Okay. So in intestine, primary bile acids undergo deconjugation and dehydroxylation and result in the formation of secondary bile acids. So your secondary bile acids, they are formed in the intestine, whereas your primary bile acids, they are formed in the liver. Now these secondary bile acids are deoxycholic acid and lithocholic acid. 99 to 98 to 99% of secondary bile acids, they undergo enterohepatic circulation. That is, they reach back to the liver. Reaches back to liver. So let's suppose, let me simplify this for you. This is your intestine. So this intestine, it receives your primary bile acid. Now what will happen? They undergo deconjugation and dehydroxylation to form what? Secondary bile acids. And these secondary bile acids, almost 98 to 99% of them, they enter enterohepatic circulation, and that is they reach liver. That is how they are then excreted from the body. So that was about cholesterol synthesis. So we finished about fatty acid synthesis and cholesterol synthesis, both of which are important. You might get asked. So the important points that you need to remember about cholesterol synthesis is the site, that is the organelle and the organs where it is taking place, then the reactions of cholesterol synthesis and its regulation. So this is the most important point that you remember, that is site, organelle, reactions, and regulation. So this is like the basic and the most important for all the cycles that you need to know, must know. Okay, they fall in the must know category. Okay, so with any queries you can ask. Thanks for watching our live. Let me see if you have any queries. Okay, thank you for watching.